Bismillahirrahmanirrahim. In today's lecture, we will discuss the cellular basis of immune response. This cellular basis it includes the various cells that are involved in immune responses, how these cells they are originated, and the various cytokines which are secreted from these cells and the effects of these cytokines. When we talk about the origin of the T cells they are formed in the bone marrow like other cells. They originate in the bone marrow, but then they are circulated to the thymus, and it is the thymus where these T cells they mature. Before entering the thymus, the stem cells they lack the antigen receptors and lack the CD3, CD8 and CD4 proteins on their surfaces. When they pass through the thymus, they differentiate into T cells that can express both the antigen receptors and the CD protein. Stem cells they initially express neither CD4 protein and nor CD8 protein, but afterwards they differentiate and express both CD4 proteins and CD8 proteins. This expression is called as double positive. Please remember, initially they do not express any kind of CD protein. That is called as double negative. And afterwards they express both CD4 and CD8 proteins, and this is called as double positive. Afterwards, the stem cells they either express the CD4 protein or CD8 protein and become mature T cells. A double positive T cell, as I pointed out, that a cell that expresses both CD4 and CD8 protein, that is called as double positive cell. So a double positive cell, if it expresses CD4 on its surface and it comes in contact with the class 2 major histocompatibility complex and the CD8 bearing cell, it will come in contact with the major histocompatibility complex type 1. So this is important in this relationship between CD4 and CD8 and the class 2 and class 1 major histocompatibility complex, that is important, and it will be continuously coming throughout the immunology lectures. Regarding the origin of these T cells, few very important processes that occur in the thymus, and that is called as thymic education. There are few important terms that are written on your screens: clonal deletion, negative selection, tolerance, positive selection, and autoimmune regulator gene. What is clonal deletion? If we talk about clonal deletion, the cells which carry CD4 proteins and CD8 proteins, or you can say that CD4 and CD8 positive cells which bear the antigen for self proteins, they are killed. And these cells which have the self antigens, which have the receptor for self antigens, they are killed by a programmed cell that is called as apoptosis. The removal of these self-reactive cells, that is called as negative selection. So this clonal deletion and negative selection, they are related to each other. This point is very important to understand because it is the underlying mechanism of autoimmune diseases. If those CD4 positive and CD8 cells which have the receptors for self antigen, they are not killed, they are not removed by the programmed cell death or apoptosis, these cells will ultimately result in immune responses against the self antigens, resulting in autoimmune diseases. But nature has created a very important mechanism of clonal deletion and negative selection, in which CD4 positive and CD8 positive cells which have the receptors for self antigens or self proteins, those cells they are killed by apoptosis, and those cells after killing they are removed, and this is called as negative selection. This is very important. This is very important in preventing the autoimmune reaction. Whole of this phenomena of negative selection and clonal deletion, it requires a gene that is called as autoimmune regulator gene. For successful completion of this clonal deletion and negative selection, the thymus or the thymic epithelial cell, they must display a wide range of the self protein. A wide range of self protein must be displayed during the maturation phase. If not all the self proteins or self antigens they are displayed to the during the thymic education, it will somehow result in development of some autoimmune disease later on at some point of life. But if all the self antigens they are displayed to the thymic epithelial cells by the thymic epithelial cells, and uh, those self antigen, the cells having the receptor for those self antigen, they will be removed. Otherwise, it will result in autoimmune diseases. Another important phenomena is the positive selection. The positive selection, it occurs for those CD4 and CD8 cells that do not react with the self major histocompatibility complex. Because for presentation of the antigens, major histocompatibility complex binding, that is important. So those cells, whether CD8 or CD4, if they do not react with the major histocompatibility complex, they are also killed. Because the point is, if they do not react with the major histocompatibility complex, there is no use of these cells in the human body. They will not be able to perform any kind of immune function. So these terms, they are important. Clonal deletion, negative selection and positive selection, they are central, they are very important. So major histocompatibility complex, it performs two essential functions. One is the positive selection and the second one is presentation of the antigen. Positively selected cells, they are removed. Only those cells they are kept which can react with the major histocompatibility complex and help in the presentation of the antigen. The double positive T cells, when they pass through the thymus, they acquire receptors which have antigenic specificity, and these receptors which have antigenic specificity, they are called as T cell receptors. Another important point to note here is that 40% of the T cells they develop in the gut. The specific portion of the tissue that is called as gut associated lymphoid tissue, or you can also call it as mucosal associated lymphoid tissue. They are very important. These mucosal associated lymphoid tissue which have the T cells, they have the intraepithelial lymphocytes, they are very important. And the level of these intraepithelial lymphocytes, they can also be raised in various diseases, for example the celiac disease. The question arises that whether the gut associated lymphoid tissue, they are enough uh, to perform the normal immune function. The answer is no. And a very common disease, not that much common though, but a common genetic disease that is called as DiGeorge syndrome or catch-22 syndrome, in which the patients they lack a thymus and they have profound immunodeficiency. So those patients who have DiGeorge syndrome, they lack thymus, but they do have the mucosal associated lymphoid tissue, but they are immunodeficient. So this is a very valid answer to this question, that the presence of these mucosal associated lymphoid tissue alone, they are not sufficient to perform the normal immune functions of human body. When we talk about the origin of the B cells, they arise or they originate in the bone marrow, they mature in the bone marrow. They do not pass through the thymus for their maturation. So as it is already pointed out in the slide, differentiate in the bone marrow but do not pass through the thymus and matures in the bone marrow. They also undergo the process of clonal deletion and negative selection. But when very important process that is specific for B cells, that is the receptor editing. What happens in receptor editing? B cells, few B cells, they bear an antigen receptor for a self protein, and they can escape the clonal deletion by the process of receptor editing. In this process, some different light chains they are introduced, they are produced, and these different light chains they altogether changes the specificity of the receptor so that it no longer can be recognized as a self protein. So remember, this process of receptor editing, it only occur for those cells which escape the clonal deletion, and they escape the clonal deletion through this process of receptor editing. Different light chains are introduced, and when different light chains are introduced, they can no longer be recognized as self antigen. What are the light chains? You people will study in the coming lecture on antibodies or immunoglobulin, that there are two types of the chains which are present in the immunoglobulin molecule, light chains and heavy chains. So if, and both of them they are covalently bound together. So this is the whole process of receptor editing that is totally reserved for the B cell. And the last important point, as much as 50% of the B cells they undergo the process of receptor editing. So this picture it shows the origin and maturation of these two types of the lymphocytes, the T lymphocytes and B lymphocyte. Both the type of the lymphocytes, they have their stem cells in the bone marrow, they originate in the bone marrow. T lymphocyte, as you can see on the upper portion of your screens, that T lymphocytes they go to the thymus, mature there, and then differentiate into CD8 and CD4 positive T cells, while the B lymphocytes they originate in the bone marrow and they mature in the bone marrow. And when they mature in the bone marrow, these B cells they ultimately give rise to the plasma cells, and these are the plasma cells which differentiate into various immunoglobulin molecules, or you can call them as antibodies. These are the few important points regarding the origin of the natural killer cells or NK cells. These are the large granular cells which do not pass through the thymus, which do not have any kind of antigen receptor and do not bear CD4 or CD8 proteins. These natural killer cells, they detect those cells which are negative for class 1 major histocompatibility complex, or those cells that do not display the major histocompatibility complex type 1. The detection process is effective because many cells lose their ability to synthesize class 1 MHC after they have been infected by a virus. So natural killer cells, they are typically important regarding the viral infections. Next is the origin of the macrophages. Macrophages, they differ from the B lymphocyte, T lymphocytes and the natural killer cells in the sense that they arise from the myeloid precursors, while all the other cells, the B lymphocyte, T lymphocytes and natural killer cells, they arise from the lymphoid precursors. The macrophages, they perform two very important functions. They are the phagocytosis and the antigen presentation. Again, the same points, they do not pass through the thymus and they do not have the antigen receptor. But one important point to note here is that these macrophages, they display class 2 major histocompatibility complex. Some of the macrophages, they also display the class 1 major histocompatibility complex like other nucleated cells. There are few important differences between major histocompatibility complex. There is a complete lecture on the major histocompatibility complex. The differences will be studied over there. So this was today's lecture about the origin of various cells which are involved in the immunology or immunity.